Thank you for joining us today. My name is Andrew Olson. I'm with QIAGEN Digital Insights, and I'll be your host and moderator for today's webinar titled Single Cell Analysis of the Tumor Microenvironment in High-Grade Serous Ovarian Cancer. Today's speaker is Jean-Noel Billaud. He is a Ph.D. senior scientist at QIAGEN Digital Insights, and before joining QIAGEN Digital Insights, Dr. Billaud joined Ingenuity Systems in 2008 as a staff scientist for in silico research programs in oncology and infectious diseases. He holds a Ph.D. in blood cell biology from Paris 7 and completed his postdoctoral work at the Scripps Research Institute in San Diego, California, where he studied the impact of the feline immunodeficiency virus and Borna disease virus on brain cells. He pursued his career at the Vaccine Research Institute of San Diego, where he co-developed a universal vaccine platform using the core antigen of the hepatitis B virus to target infectious viral and parasitic diseases and metabolic diseases and cancer. With that, I'd like to welcome Dr. Billaud.
Thank you, Andrew. Thank you very much. Today, I'm going to present you a very important use cases dear to my heart. And it's about the single analysis of the tumor microenvironment, a very serious and lethal disease, which is high-grade serous ovarian carcinoma. So before going any further, I would just like to give you a little summary of what we are talking about that. Ovarian carcinoma is a very serious disease, as I said. And we're talking about 300,000 women being diagnosed worldwide each year. And unfortunately, 200,000 women are going to die from that disease. The high-grade serous ovarian carcinoma is accounting for 70% of the death in these ovarian cancers. It's very hard to diagnose, has a very high level of recurrence. And as of now, we don't have any perfect treatment at all. So one idea is to study what we talk about the environment that allows the tumor progression. And this is about the tumor microenvironment. What is it? It's all the cells, including cancer cells, endothelial cells, epithelial cells, immune cells that allow the cancer cells to progress, to metastasize and be very efficient at what they are doing. And the role of the tumor microenvironment is to balance that. So either promoting or fighting it. And it's all about the correct balances. So a little bit of a legal disclaimer that all the products I'm going to show are just for molecular biology application only. And we are going to ask basically three questions. We are going to analyze the molecular role of cellular subset in that cancer using single cell sequencing. And mainly we can look at the cell type, which one are involved, what kind of marker we can help to distinguish this cell type. What is the biology which is underpinning that, the molecular pathways, the transcriptional program. And also we can compare what we are going to find in that single cell analysis to a bunch of other comparisons that we have in our database, and we're lucky to have that. So I'm going to present you three little teasers. And as you will notice, there is a dot on the side. And my point is to tell you how we can use our technology to cover basically all aspects of the biology that we would like to. So here it's called analyzers. I just represent some graphical summary, which is basically a synopsis of what's going on in your data set. And just to give you a couple of examples, I will detail that a little bit more. So we can analyze. We can also compare, because we are lucky enough to have over 100,000 comparison expression that came from Omixoft, which is a product that I'm going to talk about as well. We are allowed to look at any type of comparison we would like, what type of pathways, what type of activity for a particular upstream regulator, and so on. So that is the compare tool. But also we can contextualize. So once I found something interesting, what do we know about it? And we're going to use the web versions of Omixoft called Land Explorer, which is attached to IPA from the gene view link. And here we can simply look at one particular gene in one context and find out a lot of it. So that's the idea of this. Now let's go back to my agenda today. I'm going to describe a little bit what is our ecosystem, the sample to insight developed by QIAGEN Digital Insights. We are going to process the data using genomic workbench. We're going to analyze, as I said. We're going to compare as much as we can evaluate in relation about the data, and we are going to contextualize as much as we can as well, and bring you some conclusion. So the ecosystem that we have developed at QIAGEN is basically from the sample collection to the interpretations. We have a set of technology that are really well adapted. And on the far right here, we have the data analysis column and the interpretation column. Two products I'm going to talk about today, the CLC Genomic Workbench, which allows me to process any type of FASTQ file or microarray or even matrices, as I've done here for the single cells. And the interpretation will be using QIAGEN Ingenuity Pathway Analysis, which is the flagship for biological interpretation worldwide, tied with the Land Explorer, which is the tool that I'm going to allows me to contextualize the data I found, right? And I'm going to touch base a little bit of a new database that we have released recently. So what is the case we are? It's based on a clinical cancer research paper published in 2022. And they wanted to look at using single-cell RNA sequencing the tissue architecture of that high-grade serous ovarian carcinoma. So they have gathered tumor sample from patients that have done specific surgery. They have also non-malignant ovarian samples and that have underwent also some type of surgery, but we're going to use the normal part of that non-malignant ovary use. So we have tumor and normal tissues. They use 10x genomics. It's a good news because the CLC genomic workbench can process that, especially the V3 chemistry. We're going to align to B38 and quantify as normal. And we're going to give some dimension reduction using Umap. Here, I'm going to show you one case. So here it's a cartoon to simplify exactly as a biologist how I will use it, right? You have single cell FASTQ file. Here it was matrices, so think it's a matrix. I have metadata which is attached. You import that into the premium module of Genomic Workbench. You process it according to specific workflow that has been already set. It's very easy, stepwise process. And you can directly send, that's the beauty, send the data directly to IPA. And IPA, which include analysis match, I'm going to describe that, and an explorer. Now you can compare, you can contextualize, and you can explore the biology of that specific data set. Here, this is the workflow from Genomic Workbench. So here's a tag process. I'm going to process the matrix. The matrix has been used from Cell Ranger from 10x Genomics. It's a MEX format, and basically it's a workflow already set, and you process. You add the classifier of interest. Because you have multiple samples, you can iterate and collect through, and you end up to have very important information at the end: heat map, expression plot, dot plot, matrix, and so on. All of them can even do that in the cloud engine. This is the matrix I have imported. As I said, they are from the MEX format, Cell Ranger from 10x Genomics. And the protocol is basically, once you have set up your consensus genome, your gene model, which is the Ensemble V99, you know what type of classifier we use, which is the version 1.2 as of now. We are going to process all these different matrices. You have 5 of them that are normal tissues, and 7 of them that are different grade of the high-grade serous ovarian carcinoma, 2 grade 1, 2 grade 2, and 2 grade 3, stage 3. Here, this is the metadata which is attached, processed directly from the matrix information. As you can tell, you have normal tissues and cancer tissues. So I can separate everything I want, even based on age or based on the type of pathology, right? So what do we get is a Umap. And the Umap is basically here a 2D. I could show you a 3D, but it's a 2D representation of what are the cell type that are represented in that cancer and normal ovarian tissues. So here I simplify for the sake of the presentation. We have roughly 10 main cell type, and there is more. The paper I described a little bit more because they went a little deeper. I don't think it was the point here, but we have basically macrophages, T lymphocyte, natural killers, a bunch of plasma cells, fibroblasts all over the place here, endothelial cells, some myofibroblasts, some B lymphocytes, right? And a huge chunk of epithelial cell line. This is the representation as is, everything confused, normal and cancer tissues. But now I can color as well that Umap based on the staging and the normal tissues. This is the color code. Yellow, stage 1, as you can tell, epithelial, some T cells, and so on. And you have the purple and your red going for stage 2 and stage 3, right? So now we can easily look at different expression profile based on some criteria that we can decide. One of the good features that we have in CLC genomic workbench is the Variant plot. So it's basically indicating for a particular tissue, a particular cell type. Here, this is the plasma cells. We know they are B cells that produce antibodies. What are the most represented genes in that particular cells? Well, as you can tell, it's all about immunoglobulin related, heavy chain, light chain, and so on. So this is a very nice confirmation that what I find out and using the classifier as a plasma cell, it's actually a plasma cell. But we can also look at a heat map based on the cell type and the disease status. In other words, what are the most, based on 20 genes here, what are the cell type in which these genes are the most represented? So it's based on the expression influence here. The more red, more express, right? But what I would like to show you here is these three legends here. We have one for the disease, one for the cell type, and there's a color code here on the right, and one for the stage is a little bit more confused. One striking thing is for the yellow, which is the cancer type, regardless of the stages, the most prevalent cells are actually the epithelials, right? Now you look at the normal tissue, which is in blue here, which corresponds to that legend. The most prevalent cells are the fibroblasts. And that's the normal features of the ovarian tissues of women that are aging. A little bit more fibroblast is in place, right? And everything else after you have all the other cell type that are described in that legend here on that color code. So that was a one striking element that I've seen, and that confirms based on the paper as well, and regular data of ovarian tissues in aging women. Okay, we can also define a specific marker. And the way we do a specific marker is basically taking one type of cells versus the rest of the cells and compare that. What I wanted to show you is the marker for macrophages versus the rest when you are at stage 3, which is the advanced stage, the late stage of that cancer, right? In between metastasis and heavy metastasis. And what you look at that population, which I define as macrophage monocytes, again, we can probably go deeper. It's probably an M2 type of macrophages, but I'm not going there. And the marker that we have defined is, the top one is MSR1. That is a top macrophage marker at stage 3 in that particular high-grade serous ovarian carcinoma. So when I talk about contextualizations, I wanted to know a little bit more about that marker. Where is it enriched? So we are lucky to get a lot of land, so a collection of data sets. One of them is the blueprint. And the blueprint is a collection of data sets that encompass malignant and not malignant hematopoietic cells. So here I selected in which type of cells, basically, the MSR1 marker is expressed. And as you can tell, it's kind of a confirmation and it relieves the M2 and M1 macrophages. Basically, macrophages are the one that express the most that MSR1 marker. So again, when you have the process, the data, you have the expression profile, you know where it's located, you can get information based on other data sets, such as blueprint here, which come from Land Explorer. Now let's look at the differential expression analysis of that particular single cells, and you can set up any sort of comparison. The one I've looked at is based on stage, right? And looking at all group pairs. Basically, this kind of comparison. Epithelial cells, I want to look at stage 3 versus normal. I could have done for fibroblast or lymphocytes, the same things. The beauty of it is in our ecosystem, everything is tied. So once you have CLC, you have a plugin that allows to send the information to APA. And it can do that by itself, not only upload the data set and set up the parameter of the analysis. That's what I've done here. It's just an automatic process. I set up some cutoff for the p-value, for change, and you see some of the comparison I've sent. And it's directly sent to IPA. And in IPA, we can use many, many features. And what I would like to propose today, not unfortunately a key for the treatment of high-grade serous ovarian carcinoma, but more an approach to understand what type of technology we can gather to get reasonable hypotheses, right? So here, this is the graphical summary. I'm very proud of that features because the graphical summary is basically a synopsis of drawn by machine learning algorithm that will connect the most important biological entities that you have in your data set, right? Based on genes, as you can tell here, based on disease, based on pathways, and you connect all of them. And everything is done automatically. And that, what does it do? It provides you a very clear image what's going on in that data set. So I give you two examples, natural killers and fibroblast. Natural killer, stage 3 versus normal, same for fibroblast. What you can reasonably see is just looking at that graphical summary. Again, this is done automatically. I don't have any input in that. It put the pieces together. You can see that the cellular function of natural killer and fibroblasts match well together to promote that environment of high-grade serous ovarian carcinoma. Very important gene, EGF, CD40, some interferon pathways are involved, but also fibroblast, fibrosis, which is induced, and so on. So these are very nice elements and nice features you have in IPA that in matter of seconds, looking at the data and says, okay, I know what's going on now. But let's compare, because I'm lucky enough to have all these comparisons. I provide 8 comparisons here. From the left to the right, I have natural killer, macrophages, epithelial, plasma, T lymphocytes, endothelial, fibroblast, and myofibroblast, all stage 3 versus normal. And I just quickly use the comparison analysis feature in IPA and I compare all of them. And what you see is a bunch of color, right? Blue and orange. It's very simple. Orange is the prediction of activation, and blue is the prediction of inhibitions. So based on the genes that are overlapping that particular pathways of interest that you see in that list on the left here, we can predict if that pathway is turned on or turned off. So I wanted to summarize, because it could be a little bit overwhelming here, but I want to summarize by bullet point here. What's going on in that particular tumor microenvironment? You have, first of all, a general inhibition of the protein synthesis. It's special protein synthesis, but all the cell types, as you can see here, they are predicted to be inhibited. You may have also activated epigenetic and transcriptional repression, specifically in T lymphocyte, as you can see here. This is highly activated for that particular cells here. And also you have an activated fibrosis. You see that in endothelial and fibroblast. You have differential regulation of the actin cytoskeleton, particular inhibition in epithelial cells, activation in T lymphocyte and fibroblast, a decrease in inhibition of cell death. You've seen that in natural killer and T lymphocytes. You have a general inhibition of the cell-cell adhesions, especially in epithelial cells. All of that contribute to what we call epithelial to mesenchymal transitions that favorize the metastatic, right? You create a niche and you colonize that. That's the way cancer cells do. And you need all of these phenomena to happen to promote that cancer. You have a general inflammation. It's very increasing T lymphocyte fibroblast. Again, this is one of the hallmark of cancer. You have an increase in invasiveness. You've seen that a lot in fibroblast and myofibroblast. And finally, you see an increased cell survival in endothelial, fibroblast, and myofibroblast cells. So just looking at that comparison, you have a general overview of what's up in that cancer situations, where pretty much all the cells, immune and non-immune, stroma cells, cancer cells, are all fighting to each other, either to promote the environment for the cancer to thrive or to fight the cancer progressions. Now let's go deeper a little bit, and we can look at features that we have called upstream regulator. It's basically a feature that allows us to look at the transcriptional program what is going on, who is driving, who is the conductor in that tumor microenvironment. And here I have selected, because I can select many type of molecule, could be transcription regulator as you can see here, it could be cytokine, could be kinase, but I selected the transcription regulator, basically transcription factor as we know it. And I selected those that were predicted to be activated. So those that are leading a program that will allow specific phenomena, right? And I notice 4 of them are pretty important, very, very high score, KDM5A, BAC1, NANOG, and CREM. So first of all, I wanted to look at the representation in that Umap I provided to you before. Where are they expressed? So you just have to believe me, all these 4 transcription factors are expressed ubiquitously in all normal cells. That's the normal fact. But they are expressed also, as you can tell here, NANOG, in the epithelial cells. This is this cluster. All of that are epithelial cells. NANOG, BAC1, CREM, and KDM5A are all expressed in cancer cells, in epithelial cells. And now what I can do using that is I can click, it's a very simple system. You click all of that and you display one network that encompasses all these 4 transcription factors. They are there, all of them, NANOG, BAC1, CREM, and KDM5A. They are all predicted to be activated. The color is orange. And what we have done here, we have put the network in with the molecule that are associated downstream of each of these particular transcription factor. They are associated by S expression and transcription edges. And based on the expression of these genes you see here, green is downregulated in the stage 3 versus normal, or red will be upregulated in the stage 3 versus normal for epithelial cells here. Based on that expression pattern, we have determined that all of these transcription factors are predicted to be activated. Very nice score. For the beauty of the presentation here, I also asked the question, when I have a program like that of transcription factor, what type of process I can see? And as you can see, statistically significant, you have a tumor progression of epithelial cells in particular. So just these 4 guys here are promoting a big push of tumor progressions. 99 of these differential expressions are driven by these 4 guys, so must be really important. I would like to contextualize now, because let's say I don't know anything about this transcription factor. I would like to know a little bit more in relation to that cancer. Well, first of all, we can look at survival, right? So based on the expression of NANOG, what is the relation with survival in ovarian carcinoma? And as you can see, the increased expression of NANOG decreased survival in ovarian carcinoma. All of these data are tied to high-grade serous ovarian carcinoma. So NANOG becomes a marker. If you have a high expression of NANOG, you have a decreased survival. That marker is involved in embryonic stem cell proliferation, renewal, and pluripotency. So no surprise that it's associated with a poor prognosis and resistance to chemotherapy. So that's one information that if you didn't know, you find that very easy. Let's look at BAC1 and KDM5, and I wanted to see why how these guys are correlated to each other, right? And in what type of cell they are expressed. So you can look at the expression of these 2 guys, BAC1 and KDM5A, in specific immune cells. Obviously, I choose again the blueprint, which is hematopoietic stem cells, malignant or not. I selected the normal situations here. And here it's a color code of all the cells, but let me tell you, you have mainly monocytes and mainly lymphocytes. And this is based on the log2 transform of expression BAC1 or KDM5. Basically, these guys are highly expressed in both type of cells. What's important is the BAC1 is involved in the regulation of oxidative stress, which is a very key factors in progression of tumor, cell cycle, hematopoiesis, and immunity. And recently, the transcription regulator ovarian carcinoma has been shown to promote EMT for the BAC1. Let's look at KDM5A, which is a lysine-specific demethylase. Well, it inhibits the anti-tumor response in ovarian carcinoma. It's a recent paper in 2022. And it can promote also EMT. And it's correlated to chemoresistance, as we know when you use these cell lines. But I wanted to know, using a new database that we have called Tumor Mutation based on the B37 consensus genomes, what is the percentage of altered samples when you look at KDM5? Well, and you have a color code amplification mutations. Well, the KDM5 in ovarian carcinoma is heavily mutated in these and amplify in these type of cancers. So here we have three different relations. We know they are tied together, we know they decrease the survival, and we know when they amplify, they're tied to ovarian carcinoma. That's the contextualization part. Now let's try to play a little bit with the compare idea. What's up with our data versus the rest of them? And we had over the years look at combining very important piece of information. One of them is comparison expression data set. We have roughly right now 118,000 differential expression data set that come from Omicsoft. And Omicsoft has, over the years, curated normalized data set that have been public source from GeoSLA, TCGA links, and you name it. And they have processed in such a way, an elegant way, that we have control vocabulary attached with, so we can do a lot of information. This data set has been pushed and into IPA. And now we're in the features called analysis match, which I will present later. We are going to see what's up with the rest of the world when I want to query my own data set, expression data set, and see what's going on in the rest of the world. These data set encompass many things: oncological, non-oncological, single cell, and even normal tissues. So you see the power we have here. And that's just another representations, but that is important to say. Not only we combine these expression data set that has been curated, normalized, control vocabulary, but we bring also our knowledge base, which bring more than 10 million curated findings by Ph.D. experts that organize, structure the database. So the power of two together now, it's huge. So I'm going to show you three different things that has been developed in the last 3-4 years: ActivityPlot, pattern search, analysis match. We are going to look at the pattern searchers. What is it? The pattern search is, let's say you have a network, and here this is one of the case. You have some that are predicted, some that are up-regulated, some are down-regulated, but this is basically a network. You can look and query, where do I see that in the rest of the world? The world being our universe, which is this 118,000 comparison expression data set. I would like to know where do I find any match or anti-match of that particular pattern search. I would like to find that. Okay, and I'm going to give you an example. First of all, we know that the fibroblasts are prevalent in that tumor microenvironment. We know they promote invasions. We know they promote metastasis, angiogenesis, basically everything necessary for the cancer cells to progress. I have selected, using the upstream regulator, two very important transcription factors that are known to be a master regulator of EMT, TWIST1 and ZEB1. And they are represented here. I have added another one, SPDF. So TWIST1 and ZEB1 are predicted to be activated, and SBDF, which is a very important component for cell migration, is predicted to be inhibited. What I've done on the left part is I put that network, and I asked the question, what are the main functions? As you can see, the main functions are metastasis, predicted to be increased, invasion of tumor, predicted to be increased, angiogenesis, predicted to be increased, and ovarian cancer as well here, as you can say. So I know for that particular comparison of fibroblasts at stage 3 versus normal, I know TWIST1 and ZEB1 are extremely important along with SPDF. Well, I didn't know too much about SBDF, and I would like to know where is it expressed, as well as TWIST1 and ZEB1. So I'm going back to the Umap, and using a color code as well, you can tell that SBDF is mainly expressed in the epithelial cell here, so the dark blue, right? ZEB1 and TWIST1 are all expressed in fibroblasts and other immune cells. Basically, ZEB1 is a little bit more ubiquitous, TWIST1 is more in the fibroblast part here, and SPDF is uniquely in the epithelial cells. So it's very interesting because from one comparison, you do not necessarily have the transcription factor which is pertinent to that particular cell. It could be from another type of cells. That's the case here. Well, I wanted to know when twist1 is overexpressed. Again, I'm using another database which is from Omicsoft under a web-based version, Land Explorer, that you can access from GenView in IPA. It's complicated, but we can explain that a little bit more in detail. I'm looking at twist1 expression in metastatic tissue in high-grade serous ovarian carcinoma using the OncoHuman B38 gene code 33. As you can tell here, this is based on the log2 transform expression of twist1, and I look at different samples, right? Primary tumor, solid tumor, recurrent tumor, and metastatic. As you can tell, twist1 is overly expressed in metastatic, which is a confirmation of what we know, because twist1 is mainly a promoter of EMTA metastasis. So here, by non-independent sources, we can find that twist1 is effectively in high-grade serous ovarian carcinoma overexpressed in metastatic tissue. But let's go back to that idea of pattern. I wanted to know when I have, so let's go back to that particular network, twist1, ZEB1, SPDF1 network. I'm going to find out where is it, where do we find that? That's the pattern search. I have a particular pattern, up and down genes, upstream regulators that are predicted to be activated, one predicted to be inhibited, and I would like to know in what other situation that particular network is represented. So here I'm lucky enough, I have over 100,000 analyses that contain these three guys, and I'm looking for high Z-score. In other words, I'm looking for comparison that match and resemble very much what I found in the fibroblast stage 3 versus normal. And I didn't do anything here. I didn't filter. So when you click on these guys, you have that window that happened here, and it mainly shows this is in single cells without any filtering. And you look at the type of comparison, it's always cell type versus others, and mainly fibroblast. But that's my view here. But I can look and ask a question about stats and looking at evaluation of the metadata per se, so having a unbiased way. Well, that's what I've done here. This is click on evaluation of metadata, and you can see what is the most important of these 200 analyses that I've selected here. What is the most representation? Well, you can tell it's all about fibroblast. It's all about stromal cells. Fibroblast is a stromal cells. It's always in cancer situations. So the network I found in my fibroblast stage 3 versus normal is actually resembling a fibroblast signature in other cancers. So it's a very important fact, because now I know I am onto something. I can focus on that particular cell type and focus on that particular transcription factor to see what we can tweak a little bit, right? So again, that's SPDF, twist1, ZEB1 network represent a fibroblast signature in cancer setting. Could do that because I have pattern search. If not, I would not have been able to look at that. Let's look at another aspect called the ActivityPlot. ActivityPlot, it's basically, you know, I've talked about canonical pathways, I've talked about upstream regulator. What I would like to know here, in what occasion, in what condition, what disease, what treatment that particular upstream regulator, let's say, is activated or inhibited. Well, we use a feature called ActivityPlot. And that's what I'm going to do here. I selected another type of cells, the endothelial cells, which are prevalent in the tumor microenvironment, which are basically here, that cluster. And I asked, what are the transcription factors that are predicted to be activated? I selected specifically growth factor. I have a specific interest here. And as you can tell, two of them strike my attention, FGF2 and AGF. They're all activated in endothelial cells when you compare stage 3 versus normal. HGF2 and AGF, very strong Z-score. And you dig a little bit in the information, you can use the GenView, you have a lot of details and PubMed ID and papers you can go through to query a lot of information. That's what I've done, basically staying into the IPA ecosystem. What you look is FGF2 is a very important angiogenic factor. It does actually induce EMTs along with others, right, in ovarian cancer. It's known that you dysregulate its expression, it's always associated with aggressive cancer phenotype and enhanced chemotherapy resistance. Again, one of the main issues that we have in high-grade serous ovarian carcinoma is chemo resistance. Not only you diagnose very late, but it's hard the drug doesn't work very well. They work for a time and they disappear, right? And that dysregulation is associated with poor clinical outcomes. On the other part, HGF is known to promote invasion and migration of ovarian cancer cells. Its upregulation on the signal HGF-c-Met is associated with poor prognosis as well, right? I can dig a little bit further. I know FGF2, when I look at my IPA and I build the connection between FGF2 and HGF, which is not necessarily prevalent here, not obvious, we know that FGF2 increases expression in HGF, and that I can do that because I have the knowledge base under my disposal when I use IPA. Let's look at the expression of these two guys. FGF2 is expressed in the endothelial, heavily in the fibroblast, right? And I give you the proportion of these expressions. It's mainly lower expression in high grade, very high in normal tissues, but still. And HGF is mainly expressed in fibroblast, again, blue, and macrophages here, as you can tell. That's the light blue here. And the proportion is equivalent in cancer or normal tissue. So remember, we talk about the activity plot. Let's keep that in mind. So first of all, I put a network. HGF is predicted to be activated, FGF2 as well. This is the genes that are downstream of these two transcription growth factor. And what's striking as well, again, is when you grow statistically significant functions using the growth features, you see all these different type of important information. Increase of cell movement of epithelial cells, cell proliferation fibroblast, migration of endothelial cells. Remember, the comparison that I've seen here at the beginning was endothelial cells, and you can get information for other type of cells, including cellular infiltration by leukocytes. You promote EMT, you promote metastasis. Generally, you promote migration of tumor cells, and you decrease apoptosis. Very important information you have with these two transcription factors. Well, what I would like to know, because let's say I didn't know too much about HGF, and I wanted to know what's the relation with the HGF and TP53. Why TP53? Because this is the most mutated gene in ovarian carcinoma. 91% of the case, according to TCGA, we have a mutation of TP53. So that tumor suppressor is mutated. And here you can see the expression of the HGF based on the status of mutation of TP53, not status, but expression of TP53. Its HGF is highly expressed when TP53 is down, right? And it's inversely correctly when it's lower expression when it's up. So everything works well. So in a situation where you have high mutation, very low expression of TP53 in ovarian carcinoma, IGF is up. So again, it makes sense to see at that network when you look at IGF and IGF2, right? I can ask a couple of other questions before I show you the activity plot. Is the mutation data of HGF, right? Again, I didn't know anything, and we have that tool and Explorer, which is tied to IPA. Well, I can look based on the stage, the clinical stage, stage 4, 2C, and 3C. What is the type of mutation you have for HGF in ovarian carcinoma? As you can tell here, a lot of implication when you're in stage 3. Everything I've shown you so far is stage 3 versus normal. So we have some kind of relationship, which is HGF amplification and ovarian carcinoma here. I can even look using a genome view, the type of DNA-seq mutations of these HGF according to the different stages, and you have a color code for the type of mutation. So it's very useful. Finally, I would just like to touch base with something that is really important in our ecosystem, HSMD, looking at basically clinical data of HGF across cancer for that particular genes. Well, HSMD, Human Somatic Mutation Database, is new, right? It's newly developed by QIAGEN and has content from knowledge base, which is expertly curated content, and also data from real-world oncology cases from our professional clinical interpretation services. And what it does is quickly I query HGF, and you can have on the bottom, it's a small screenshot here, but the type of alteration you find for HGF across cancer, right? What type of functional impact you have on the particular protein, the actionability, which is, the clinical significance, tier 1, tier 2, according to Umap, ASCO, and so on. You have that quickly. And on that one, you have a tier 2, which is potential clinical significance for when you have an amplification. I click on that, and you can look at the type of clinical trial, what's going on for that particular amplification of HGF, when you see the drugs, the specificity of the profile of HGF, when it's positive, when it's overexpressed, amplify, and the different clinical studies you have for the different cancers. It's very easy, very, very simple to use. But let's go back to ActivityPlot. I'm asking the question, when does HGF is increase? So I specifically asked the question, when is it increased in cancer? So I selected the database called OncoGeo, again, come from Land Explorer, which is the web version of Microsoft, and that's the only filter. This is all these bunch which are in green that are very high Z-score, so showing that HGF as an epsilon letter is predicted to be activated, and this is the bunch of data set we have. Simple. I'm going to look at the metadata. When does HGF activity is up? Well, guess what? HGF, when you look at disease state and treatment in tissue, you realize that HGF is active, is increased in presence of HGF2, as you can tell here, in very high percentage. So you have a strong association of HGF activity and HGF2 activity. So therefore, the network I've seen before, which I didn't know anything about, I can find out this is a known phenomenon across cancer, or at least highly prevalent. See, that's the beauty of having ActivityPlot. And finally, let's look at analysis match. And what is it, analysis match? You know, we have all these data sets. I talk about 118,000, and for each of them, coming from Microsoft, we have calculated the Z-score for the canonical pathways, for the epsilon regulator, for other things I didn't talk about, such as the disease and functions, and so on. And we have a general Z-score. And basically what we are providing for each of these 118,000 data sets, a value of Z-score for the canonical pathways, the epsilon regulator, and so on, right? And like that, you can see if what your own data set you're going to query against have any similarity with any other data set in what context. Let me give you an example. But first of all, I would like to show you a very nice representation we have right now for the canonical pathway. It's a bubble chart. And I just wanted to show you customizations. I have the pathway category that we have in our IPA. We have over 750 canonical pathway already preset with different categories, as you can tell, growth factor category, cell cycle, nuclear receptor, and so on. And I put on the x-axis the Z-score. So basically, what you see here is orange and blue. First of all, on the left, it's blue. So every pathway that are predicted to be inhibited. On the orange, every pathway that are predicted to be activated. But most importantly, the size also corresponds to the number of overlapping genes that are in that particular pathways that allows the calculation of the Z-score. But what I wanted to show here, this is about tinafocyte. This is the comparison where I'm looking at tinafocyte, stage 3 versus normal. And you can tell there's a lot of things going on in the cytokine, cellular immune responses, cellular stress, heavily orange, and other things. So the bubble chart will give you a very quick glance also of what's going on in your data set. Obviously, you can click on any of these bubbles and find out what is the pathway. You have a report if you don't know what that pathway is talking about, what are the genes, and so on. But let's go to the match. So I'm looking at my question is, when I have that tinafocyte stage 3 versus normal in high-grade serous ovarian carcinoma, I would like to know if there are any similarities or dissimilarities to that particular comparison in other cancer. That's what I've selected. So I selected the oncolon, which include all comparison expression in oncology, colon, and I look at treatment versus control. I have a reason behind, because I would like to know in what conditions that stage 3 versus normal for tinafocytes, does it bring anything of value for me? Is there any particular treatment that it represents, or is there against or dissimilar of treatment that could be useful further after, right? So I selected that. There's 200 analysis being selected. And what you can tell when you look at the metadata, again, I look that evaluation of the metadata, it's very clear. The majority of the treatment include interferon, right? Whether it's gamma or alpha. So when you select these conditions, I want to look at tinafocyte stage 3 versus normal. In all cancer, and I look at just treatment versus comparison, it's all about interferon, all about that interferon, and very high Z-score, as you can tell, for the upfront regulator. Well, let's find out. So now I put these different comparisons that I've selected here, the one you have seen, the 10, 12 you've seen here. I select that and I project a heat map. That's the heat map you see on the left, right? And that is T lymphocytes at stage 3 versus normal in comparison to many other data sets. And as you can tell, as quick things, when you select upstream regulator as the entity here, there's a clear program of activation and a clear program of inhibition for some of the upstream regulator and others, right? And the game right now is to understand what's going on here. What's going on? Well, we have the metadata, as I said, that comes from Microsoft because they have carefully controlled the vocabulary. And therefore, we can see what's going on in each of these different headers here, which correspond to one particular data set. So I click on each of them, and I found out a lot of information. And the information I want to resume is that one, right? Two aspects. First of all, the metadata seems overall indicates that targeting other genes may mimic the addition of interferon for raising an anti-tumor response. And I'm going to put that very lightly because anti-tumor response, I can defend that aspect. I can also defend the pro-tumor response. Again, I'm talking about a balance here. The fight to get rid or promote the cancer is really tricky. And all the cells combined do that. But that's what I found here for the metadata. First of all, FGF2 receptor, which is the receptor of FGF2, activating mutations have been seen in breast cancers. And they are in combination of another estrogen drugs and a MAP kinase 1 inhibitor. It does suppress the chemoresistance to fulvestrant. So very important. So that activation of that particular guy is important. When we inhibit one of the components of the polycomb repressant complex, PRC2, one of the components is EED. When you inhibit that component, you induce regression of tumors engraft in vivo. And we have seen that in non-Hodgkin lymphoma. We find out also that when we have a copper chelating drugs that promote the PD-L1 degradation, remember PD-L1 is used by the cancer cells to promote their life, right? Their cycle. When you promote that degradation, you increase the number of CD8 infiltrating cells, which are very important and natural killers. By doing that, you slow the tumor growth, and it has been seen you improve the mouse survival of neuroblastoma. We find out also that when you have GSK3 inhibitors, you allow the Wnt activations that reduce the formation and improve the survival in another type of cancer. We find out as well that we have MEK inhibitors that counteract the interferon resistance, which is observed in CML, in kidney cancer, and fibrosarcoma. And finally, when you degrade, and it's very important I show that to you here, when you degrade that particular compound called Icaros, which is by acting on E3 ubiquitin ligase, you induce T cell activations, and you induce apoptosis and growth inhibition in malignant B cells and other diffuse large B-cell lymphoma, right? So just by looking at metadata, you have that type of information. For the scientific prospect, I wanted also to show you in red and in blue what you also found looking other type of data you may have in IPA. You can see the interferon pathways, as I say, seems to promote an anti-tumor responses, but it can go both ways, right? It depends on the cancer, it depends on the situation, but interferon gamma can be an anti-tumor or a pro-tumor, again, depending on conditions. The interferon type 1 here, as you can see, and IRF1 are very anti-tumor in general. But you see in the same heat map, FOXC1 and prolactin and a PRDMS, which is at the same time activated or inhibited, are promoting the pro-tumor environment. It's a very complex system. But what I wanted to show you here is with IPA and Explorer, you can have an overview of everything that can be very useful to understand and bring new hypotheses in context. And I would like to conclude, right? First of all, we can identify biological clusters, very important. We can see this is an interplay with many cells of immune and non-immune origin. we can look at chemical pathways. We can look at upstream regulators. We can see what are the different programs that are in play. And we can look at cell-specific signatures and try to look from the rest of our universe, which is this huge set of data sets, which is basically hundreds of years of work at our disposal. We can compare and try to understand what's going on in your data set. On that note, I would like to thank you very much, and I hope you have appreciated that little presentation. Thank you.
Thank you, Dr. Billaud, for that wonderful and interesting talk. We do have a few questions that we'd like to send your way and address those. First question is the Umap plots that you showed throughout your presentation. Do those come from IPA or do they come from something else?
So Umap is representation of what they call dimension reductions, and has been provided by CLC Genomic Workbench. As a matter of fact, you process the matrix of FASTQ file. One of the output is the Umap directly. So it's very easy, and you have that representation in 2D or 3D. So CLC in that particular situations.
Great. Okay. And then another question: can the general processes for comparing and contextualizing be applied to normal bulk RNA-seq data, or is that only for single cell data?
Absolutely, for any type of data. This is an example of single cell because it's particularly trivial, particularly complicated, but we have done that many times for bulk RNA-seq. Yeah, absolutely. Microarray, anything you can imagine, even proteomics.
Great, great. You did 8 pairwise comparisons in IPA. How many comparisons can IPA handle at once? What's the limit?
So for the visualization, I put up to 20 here, just for my laptop real estate, 20 comparison here. Then you can play with the different other comparison to make some kind of a bigger statement. But here is 20.
Okay, great. Thank you.
So in the match, you can select basically 100 of them, right? But for the visualization itself, it's 20.
Got it. Thank you. Are all the visualizations, heat maps, networks, etc., exportable from IPA?
Yep. You can JPEG, PNG, and TIFF. Yeah.
Great. Do all IPA licenses have access to analysis, match, and pattern search and Land Explorer?
I would like to refer to the Global Product Manager and the sales, but IPA now came, I think, analysis, match, and Explorer. It's an addendum, I think.
Right. Okay. And then a slide-specific question. Did the data that you presented on slide 22 come from the data set you analyzed? I'll let you get back
to... Go back to the 22.
Yeah. Yeah.
This one?
Yes. Did the data that you presented here come from the data set you analyzed, or is this generalized data from another source?
So this is generalized data from another source. So I explained that Omicsoft has curated and normalized thousands of samples, make comparison expression data set, and they are gathered in structure that we call oncoland, diseaseland, and many others. These OncoGeo blueprint and tumor mutations are some of the lands that we have in that Omicsoft. That has been accessible in Land Explorer, which is the web versions that we can get direct link from IPA GenView. So this is external data, and that's the beauty of it, because now it's unrelated to my own comparison expression and give real information in cancer situation, in T-cell situations, or what is the tumor that we have in that particular genes. So very external, very useful.
Great, thank you for that clarification. Okay, I think that brings us to the end of our question and answer session. Again, I'd like to thank Dr. Billaud for his wonderful presentation and making himself available for our questions and answers. And that concludes our presentation for today. Thank you.
Thank you. Thank you all.